I will be starting off here by giving a quick overview for members from the microeukaryotic, the microeukes group on what our working group is up to, and then we will hear from the microeukes group on what they have been up to, and then from there we'll go into breakout groups with discussion questions. So for all the Mixotrophy working group members, this will be very familiar. So our working group goals, as they were spelt out in our proposal, was 4 of them. I just copied over what we have written. And first one was synthesize currently available data on in situ mixotrophic activity or presence, and the methods used in peer-reviewed literature. So kind of doing a synthesis paper on currently published data. Number 2 was to outline the most pressing research questions related to mixotrophy. Number 3 was to then develop guidelines for the best application of current methods used in the research of mixotrophs in the environment and identify new methods for advancing potential in situ based research. And then the 4th one was to chair an ocean sciences session. We're working on this. So this is kind of an in-process thing here, and I won't be talking much about that today. We did this for the 2022 OSM, so there's really not much to say about that. It was a very specific thing. But these other 2 goals, so in terms of outline the most pressing research questions related to mixotrophy, we actually did this at a meeting in October 2021. We had a lot of discussion around this and narrowed down to 5 research priorities, and then we wrote a white paper based on those research priorities that I think we're pretty proud of, and that paper is currently in review at L&O Letters, so we kind of have our fingers crossed on that. And this is just the first round of review, so we'll see how things go. But these specific research priorities are evolution of mixotrophs, so looking at the fact that the active or the trophic mode of mixotrophy is present in many different lineages across just the tree of life. And so it's, kind of evolved multiple times over, and looking at these evolution, so looking at the evolution of these mixotrophs, we're hoping to understand common ecological pressures that's selected for mixotrophy, and helping to understand the past and the long history of mixotrophs could help us better understand the future of mixotrophs and the trophic mode of mixotrophy. From there, we talk about mixotrophy trade-offs. So, being a mixotroph, you have access to 2 different types of nutrient modes, which is really beneficial. You should never go hungry under those conditions, theoretically, but not every cell is a mixotroph. So there has to be trade-offs associated with being a mixotrophy, and really understanding those trade-offs and these dynamics of the benefits of being a mixotrophy uses mixotrophy versus an organism that's just using strict phototrophy and heterotrophy is really important. It's understanding the conditions that favor mixotrophs. The next one was understanding biogeography and ecological determinants of mixotrophy. So this is just really understanding when and where you would expect mixotrophs to dominate in aquatic environments, both in time and space, and also the conditions of which those mixotrophs would actually be engaging in mixotrophy and using both the nutrient modes, and understanding what are the environmental factors or the biotic or abiotic factors that favor then the presence of these mixotrophs and the act of mixotrophy, to understand their position in the global ocean. And then kind of once you understand where where they're expected to dominate and when they're expected to dominate in aquatic environments. The next thing to understand is then what's the impact of those mixotrophs in these locations, and that's our fourth research priority: biogeochemistry and trophic transfer. So understanding just nutrients that are being taken up by the mixotrophs, which nutrient mode they're coming from, and then how those nutrients are being transferred throughout the ecosystem, whether it's up to higher trophic levels within the microbial loop, being sequestered, what have you. And then the final research priority is this emphasis on in situ detection of mixotrophy and mixotrophs, and specifically being able to study mixotrophs in the environment. Just the way research has been conducted was primarily assuming that you had phytoplankton and zooplankton, and so methods were developed to target those two different groups. But with mixotrophs, they're this third group that often get lost within the groupings of zooplankton or phytoplankton because they haven't been targeted specifically. So how do we come up with methods that we're actually able to isolate and target mixotrophs? Because our methods weren't designed to do that. So now where our focus really is this idea of developing guidelines for the best application of current methods, and used to do the research of mixotrophs in the environment, and then to identify potential new methods. And so we're actually working on a paper right now that outlines the first four of our research priorities here, proposes potential questions, but then goes over how you could use different methods to address these questions. And a lot of the methods that we're talking about are things like BRDU labeling experiments, fish probing, fluorescent particle consumption, food vacuole staining, imaging cytometry, transcriptomics, panfluorometry, stable isotopes or radioisotope probing. And we're really, really interested a lot in this imaging cytometry or just flow cytometry in general, in situ transcriptomics, and the use of stable isotopes. But more importantly, it's really about studying mixotrophs using a combination of all of these different methods, because using just one method can only tell you so much, and finding the appropriate combinations of these methods is likely what you need to really get at what you want to address. I just want to end here on this figure that we put together for our white paper, just kind of an idea of a dream cruise where every scientist on the research vessel is doing something related to collecting data on mixotrophs or mixotrophy, and fully understanding just multiple aspects of their presence, their activity, and then their role within the food web. I'll end it there. I don't know, Becky, do you think we should take time for questions or just move on to the next one?
Does anyone have any questions?
Or anything they want to add to what I said that you think I overlooked from our own group?
Okay.
All right.
So I see that Chuck Delwich has joined us. I don't know if he prepared a short presentation on the Microeuks group. I can give a very short overview if you would prefer.
Becky, it would be awesome if you would. I had
Okay.
no idea that I would be expected to do that.
I didn't make slides for it either. So Nicole made some great slides, but I'll just briefly introduce what the Microeuk group has been up to, or what they were established to do. Back in the summer of 2021, this group was formed because there's a huge amount of research being done on microeukaryotes, but in greatly different fields like parasitology, oceanography, evolution. So what we wanted to do was to try, well, what the group was trying to do was to establish a community for these researchers and to help define some community research priorities. And back in June, they had a workshop, a 3-day workshop that focused on community building and community priorities. And from this, they came up with 7 different priorities. One was produce diversity. The other was revising the principle of ecology. A third one was similar to our Mixotrophy, is a toolkit for in situ ecology of microeukaryotes. The other was understanding interspecific variation. The other, genomic and transcriptomics. The 6th one is evolution of morphology. And the 7th is the cataloging of various taxa. These are all available, information on them is all available on the group's Slack channel, which you can gain access to by contacting Laura Katz. I believe she's still the manager of that. If I'm incorrect, please, please let me know. From this, the group has also tried to establish, put together some interactions between PIs to do things like write proposals and workshop or meeting proposals. And from that, they've written an NSF BII proposal and most recently submitted a Gordon Conference proposal. So if you have anything to add to that, Chuck, please do.
Well, not really, other than to say that we did submit a Gordon Conference proposal, which we should hear about in a few months. I've actually now forgotten when they said. I think September, October.
Okay.
So I think we might hear fairly soon. But that was built around understanding transitions in micro-euc lifestyles. So it's a very broad theme, with the idea being that each meeting would be a little bit more focused, but there would be a lot of opportunities for good meetings built around different concepts, such as transitions to and from mixotrophy, just to name one of many possible themes.
And that's, so the very diverse nature of the microeuc group is why I wanted to try and get both of our groups together to perhaps help build some collaborative bridges. And I know that there are quite a few people in the Mixotrophy working group that are also members, besides Holly and I, of microeucs. But I had hoped that this would start to open up some other conversations with people doing eukaryote research that could potentially help to broaden our Mixotrophy research goals. So that's why I tried to get us together here. And I came up with 3 discussion topics for us to go through in a little bit. But I thought it was suggested to me that it would be good if everybody took about a minute to introduce themselves to everyone else. There are a couple people here, I think, that we don't necessarily know. So think about, for a couple seconds, your 1-minute description of what you do, and we'll start going around and having people do that. I can start off. I am a molecular microbial ecologist. I study protists in the marine environment with a focus on Antarctic and sometimes the Arctic. And I am interested in all forms of mixotrophy. I'll pass it off to Nicole.
I figured it'd be next. I am a phytoplankton ecologist at the Virginia Institute of Marine Science in Virginia. And I I'm an assistant professor, so I'm trying to get a lab up and going, which feels like I've been here for 2.5 years and I should have that up and going, but considering COVID really delayed all of that, I'm a little bit behind on being successful with that. And I'm also interested across the board in a lot of different topics of mixotrophs and mixotrophy, by the fact that I've got 5 proposals I've submitted currently, waiting to hear back on, all related to mixotrophs, but all targeting very different questions related to them, from studying like copepods, eating them, looking at using stable isotopes to study the uptake of carbon from different nutrient sources, and using um, uh, uh, qPCR to study or to estimate abundances of mixotrophs in previous samples, things like that. So all over the board. And while I don't have any official projects going myself, I have got students, at least one who's working on mixotrophy and I've got new students. I'm hoping to get at least one of them interested as well. Oh, and I'm also one of the leads of the mixotrophy working group. I will pass it on to Charles. He's next on my screen.
Hey, so first of all, if you managed to get 5 proposals out, I think you're doing great. So I'm Chuck Dulwich. I'm a professor in cell biology and molecular genetics at the University of Maryland. I come out of a botanical phycological tradition, and I'm interested in, I guess I would say, major transitions in the early evolution of eukaryotes. So going backwards in time, I have tended to work on origin of a terrestrial flora from green algae, endosymbiotic origin of chloroplasts, and integration of the plastid genome into the eukaryotic genome, and then broader questions having to do with eukaryote phylogeny and the origin of eukaryotes. And let me see, who should I pass it on to? George, how about you?
Sure, I'm George McManus at University of Connecticut. I've been working primarily with ciliates that retain the chloroplast from the phytoplankton that they eat, although at an earlier stage in my life I did work on these little tiny flagellates as well. And I'm also co-chairing a SCORE working group on mixoplankton with Aditi Mitra at the University of Cardiff in Wales. And I will call on next person up is Iset Atikan. Sorry if I butchered your name.
Well, I'm retired, so
okay.
most of my work has been on cell biology and the fluidity of the membranes, mainly of HeLa cells. And then I jumped ship and tried to become a marine biologist, and that's where my interest in protists has emerged. And right now, all I'm doing is reading and listening to you folks and learning. That's it. That's all I am.
Okay, Matt, why don't you go next?
Great. Hi, I'm Matt Johnson. I'm at Woods Hole Oceanographic Institution. I study marine protist ecology and evolution. Mostly I study the mixotrophic ciliate Mesodynium rubrum, but I'm interested in a variety of other mixotrophs. And I'm part of both groups, although I haven't been present at a Microeukaryote meeting in quite some time. Sorry, I'll pass it off to Jessica.
Hi, I'm Jessica Liu. I am a research scientist at NOAA Geophysical Fluid Dynamics Laboratory. I'm a marine ecosystem modeler and biogeochemical modeler, and I'm interested in large-scale dynamics of plankton and marine ecosystems as it relates to how climate impacts mostly. And yeah, I'm one of the co-chairs of the Mixotrophs and Mixotrophy Working Group, and broadly interested in modeling mixotrophs. I'm gonna pass it on to Ariana.
Hi, sorry, I was not also not expecting it. Hi, I'm Ariana. I'm a PhD student in the MIT Who We Join program, and I have not been to one of these meetings before either. I'm just here to sort of listen in. I primarily work on coccolithophores in terms of my experimental work, and then some sequencing, lab transcriptomics, and then working in the field with metatranscriptomics. Primarily interested in mixotrophy right now from the perspective of trying to identify mixotrophy in situ from an omics perspective. So trying to look at functional differences in the expression of genes of organisms that can switch or maybe mixotrophs in different environments. So we're primarily looking at that in omics data using machine learning methods at the moment. So yeah, pass it to, I guess, Steph has not gone.
Thanks. Stephanie Dudkiewicz, I'm at MIT. I'm a researcher. And like Jessica, I'm interested on the global scale modeling. And a lot of the global scale models don't have mixotrophy, which is almost like horrendous when we think about how important they are. So I'm sort of here interested in learning more so that it's something we can put into the global scale models in best manner possible. And let me see that. Should we keep in the modeling thing? Did I pass it on to Susanna?
Thank you, Steph. My name is Susanna, and I am a postdoc at the University of Southern California. And I am an ecological modeler as well. And I've been thinking about how modeling mixotrophs and how they impact ecosystem function. But also more recently, I've been using proteome models. So really thinking about a cell and what the cell should do, and thinking about how we could couple these kind of models with omics data sets. And now I will pass it on to Erin Jones.
Hi, I'm Erin Jones. I'm a PhD candidate at the Graduate School of Oceanography at URI. I study microzooplankton grazing using metabarcoding and metatranscriptomics methods. Basically just to understand who's there in the community, how they're structuring it based on their feeding mode or trophic mode, and if I can identify grazing related genes using omics methods. Oh, and I'll pass it to who hasn't gone. Ashley?
Hi, everybody. My name is Ashley Maloney. I'm a postdoc at Princeton University, and I'm an isotope geochemist. And I'm very interested in how microbes impact the isotopic composition based on changes in their metabolism. So that's where mixotrophy comes in. It's a great way to look at a variety of metabolisms in one organism and see how its own stable isotopes change. The big dream there is to find relationships to potentially reconstruct metabolism of the past, whether that be mixotrophy or infection, or just generally heterotrophy versus phototrophy in lakes and oceans. Yeah, and I'm one of the mixotrophy working group members. And I will pass it off to, let's see, Karen. I don't think you've gone yet.
Thanks. No, I haven't. I'm Karen Stamiashkin. I'm a research scientist at Bigelow Laboratory for Ocean Sciences in Maine. And I'm a zooplankton ecologist, as well as a kind of carbon cycling biogeochemist. And I'm really interested in how mixotrophy ecology impacts nutrient cycling in the ocean, and also how they change the transfer of energy going up the food web through trophic interactions. I am also I'm a co-lead of this group, although I've been somewhat absent, so I'm happy to be here today. Is there anyone else that hasn't introduced themselves that would like to? Pat, I think, have you introduced yourself?
Hi, I'm Pat Glibbert, University of Maryland Center for Environmental Science. And I'm interested in all things plankton, particularly HABs, nutrient cycling, and particularly how mixotrophy feeds into HABs, and the balance between mixotrophy and inorganic nutrition, and how we can model that.
I think we have two people still. One is Suzanne.
Yes, hi, I'm Susanna Wilken. I'm an assistant professor at the University of Amsterdam, and I'm a microbial ecologist, so I'm interested in well, interactions in the microbial food web, and in particular mixotrophy in both freshwater and marine environments. And I combine laboratory experiments where I look at detailed physiological and molecular responses with fieldwork where I'm trying to quantify food web interactions. So I'm also using stable isotope approaches and various approaches in the field. And who else has not gone yet?
I think
He hasn't.
Margie.
Oh, yep. I was being stealthy there. I'm the mysterious MM. But I'm Margie Mulholland, and I'm a professor in oceanography at Old Dominion University. And I study various aspects of nitrogen and carbon cycling. And at present, I've been working on mixotrophic dinoflagellates in the Chesapeake Bay. And with a lot of graduate students, we've been trying to work on methods to quantify both grazing and osmotrophic use of organic compounds in natural systems. And like Pat, interested in all things nutrient and plankton.
Wonderful. Thank you, everyone. There are a couple in the chat for people who aren't able to actually talk on the because they're in places like trains. Cynthia, Heather, and Mai have put in the chat their their jobs, their interests. We do have 3 breakout rooms set up for people to go to. You can choose which one you want to go to based upon the topic for discussion. Normally we just randomly assign people and everybody discussed all 3, but I thought that this time I'd allow people to pick which topic they initially wanted to talk to people about, and then switch between rooms. So we'll see how that goes. Well, I think this might be everybody. Little attrition. So, but thank you everyone for joining the groups. What we normally do at this point is have a short recap of the discussions from each of the groups. I can start that off if you'd like. It'll be relatively brief. These will also be posted on, I believe, do we post these online, Nicole? You're muted, Nicole.
Sorry. Do you mean the notes or the recordings?
Well, we post the recordings. Do we post the notes?
We haven't really posted the notes. They've kind of just been on the Mixotrophy working group Google Drive. And I think maybe for one meeting I sent around summaries that I did, but I think I fell off the wagon on that. So
it's okay. Well, I can give then a quick overview of the using mixotrophs as model systems to study life transitions. Basically, what we discussed in there was that constitutive mixotrophs and non-constitutive mixotrophs can provide us a view of kind of different lifestyle transitions. So the non-constitutive mixotrophs allow us to study different levels of incorporation, whereas the constitutive mixotrophs could allow us to study the different stages of the transition. So going from being strictly phototrophic to being mostly heterotrophic. And in terms of actual model systems to do this, the non-constitutive mixotrophs, we're looking at either a dinoflagellate, a ciliate, or a rhizarian. And again, they offer different views. So the rhizaria are probably the least incorporated in terms of being able to control the taken up chloroplasts, and then ciliates and dinoflagellates have various levels and various means of doing that. So it seems that dinoflagellates
we lost Becky.
What, Nicole?
We kind of lost you there. You froze up for a second.
Oh, sorry. But with the non-constitutive mixotrophs, one of the issues is that they basically have wonky genomes, which makes understanding how they do things more difficult, or even getting genomes more difficult. So ciliates have the dual genome issue of micro-macronuclei, and dinoflagellates are basically a trash can for everything they've come across in their entire life. Transcriptomes can be helpful, but sometimes they don't. It can be hard to get the taxonomy of the things that you're looking for. Ciliates also do some weird things with editing, so it makes it a little harder to understand how their genomes have evolved. But we do now have the tools to start doing that. And people are starting it with mesodinium, so we'll start to see how this all works out. As far as constitutive mixotrophs, it seems that Ochromonas is starting to take the stage as being one that's within that genus. You have all these different stages. It goes from being almost an entire phototroph to being a heterotroph, relying differently on mixotrophy. It's really easy to grow, and it has also fresh and marine taxa. And in terms of genomes, it might actually be a reasonable genome to at least look at and potentially to start manipulating. So if anybody can come up with a less strange non-constitutive mixotroph, that would be great, but that doesn't seem to be the case. So would anyone from the other two?
Erin, you kind of were group 2 if you want to go next in order.
Oh, sure. Thanks, Jessica, for writing notes. I actually wrote all my notes on paper and hadn't written them on the doc yet. But so our group was supposed to be about traits and trade-offs, and we kind of deviated very quickly away from that to talk about mixotrophs in large-scale modeling and observational data sets. I asked a question about just how people's thinking had changed or not changed over the course of the last several months in light of all the writing and discussing and reading that we've done. And so one statement was just that we all have a greater understanding of how complex and challenging this topic is, which I don't need to hash that out again. But then Jessica actually proposed to a working hypothesis. Jessica, do you want to introduce your working hypothesis since you're right here? I don't need to speak for you.
Sure, but I'm sure you would have done a great job too. I was just thinking in the past couple months that one working hypothesis for when and where mixotrophs could be most important is particularly in the timing of blooms. And where this would be most important would be in, for example, HNLC regions where there's micronutrient limitation, where mixotrophs can augment their nutrient limitation by phagotrophy, heterotrophy. Yeah, so then this led to a bit more discussion.
Yeah, so Pat, I think Pat bounced on out of here. She probably got hungry for lunch or something. But so she was talking about her observations of Carenia brevis in Florida, I believe. And she said that basically under nutrient or nitrogen replete conditions, the diatoms kind of overtake the mixotrophs, but that the timing that the mixotrophs bloom is related to those nutrient conditions. And then when following that bloom, she has observed a decrease in picoplankton. They think the mixotrophs are taking down the picoplankton. So she's actually been able to observe this rise and fall of these different populations based on top-down and bottom-up effects. And then Susanna pointed out that in her modeling work for the L4 region off of the never know the British Isles. I never know what to call them. Um, she has seen that that it's not the timing necessarily of different phytoplankton populations, but it's the magnitude of the diatom bloom that's impacted by the introduction of mixotrophs into a model, that the larger mixotrophic plankton graze down these diatoms. But that for nanomixotrophs in her model, bloom timing is related to their trophic strategy. Am I getting that right, Susanna? Yeah, okay, thanks. And then we talked about the Darwin model. So Steph was able to chime in about her global model. And she did not see a big difference when she was taking mixotrophs out of her model, but it wasn't focused on the phenology or timing within a seasonal cycle of the mixotroph populations. It was focused on annual averages. So that seemed to be an interesting place that one could delve into deeper. Just reading our notes here. Oh, she did point us to a paper that Andrew Barton wrote in 2013 that got into the succession of different sized diatoms and flagellates, and saw potentially evidence that there was like an interplay of the timing of those different populations in relation to size and trophic strategy. Oh, and we talked about what would be our dream study if we could do one experiment now, our dream study. And so Steph said that her dream study would be for us to do a bunch of measurements of mixotrophs. This isn't like one experiment, but do a bunch of measurements in several key locations over several months to really nail down the things, I'm assuming, to nail down the environmental conditions that are driving the patterns of mixotroph populations in different locations, and that the timing would really be key because that could give us some important information about how to tune the impacts of environmental factors on mixotrophs to know like when they're blooming or not and crashing, etc. Group, does that sound about right? Okay.
All right, so I will go. Actually, I'll start where our discussion ended, because where we ended discussion probably where we should have started our discussion, and that was with I posed the question of like what data or types of questions. Oh, sorry, I should mention right away. Our group was talking about environmental measurements of mixotrophs and in mixotrophy and like possible approaches or methods to accomplish that, and the questions that we thought, or the types of data and the questions we thought were most interesting to us, probably biased by the people who are in the group, but within these, mixotrophic communities, there's mixotroph communities, understanding the balance of phototrophy and phagotrophy, what's triggering a shift in those balances, and then how the mixotrophs impact the nutrient balance of the whole phytoplankton community based on the mixotrophic activity of that community. So that's when I say the nutrient balance, that's like the balance of how phototrophic versus how heterotrophic the community is. And then also really emphasizing what specific nutrients mixotrophs are taking up from their different nutrient modes. So are they getting carbon, nitrogen, phosphorus, trace metals? What are they grabbing from each different mode that they're using? And so with those types of questions in mind, the different possible methods we discussed kind of covered a lot of range of topics, and I started by putting Margie Mulholland on the spot because she had said that she was had been interested in this types of stuff in her laboratory, and so she was talking about her laboratories or different students that she's working with are doing a range of different things that includes trying to culture a dinoflagellate heterotrophically, and then comparing the transcriptomes of that heterotrophic version of the culture to an autotrophic version of that culture. It's like just a culture that's just there to be a heterotroph. But of course, running into issues with the fact that, a lot of these species do require some amount of light to grow. So it's hard to grow them strictly heterotrophically. So they're also trying to reduce down the nutrients a whole bunch and maybe trying to get them to grow heterotrophically, not by removing light, but by removing nutrients. Also looking at, running more variations on the dilution methods to try to get at mixotrophs, in terms by combining the dilution experiments with imaging cytometry. So you're running the dilution experiments, and then these samples are being analyzed through imaging cytometer. So you're able to specifically not just analyzing the whole community, but specific species within that community, looking at the growth, the growth rates of and the grazing rates on these specific, all these different species you're able to analyze. We talked a lot about stable isotopes in the environment, which is a popular topic within our working group, and different ways to use these stable isotopes, and how to actually, once you enrich a community with stable isotopes, how do you then target mixotrophs, using single cell sorting. Ashley was talking about work that she was doing with trying to target nutrients going into lipids and fatty acids, things like that. Another thing Margie talked about was culture independent growth rates. So within the natural community, trying to tease out how much of a growth rate is attributed to the photosynthetic growth rate versus the heterotrophic growth rate, and get at mixotrophic activity that way. And we also then talked a little bit about using the lysotracker to stain food vacuoles. George was also brought up the idea of using electron microscopy to look at the food vacuoles. And as I understand, the evolution of, or not necessarily evolution, but the use of food vacuoles within the community. If you want to add a little bit to that, George. Oh, you're muted. Sorry.
Yeah, if we knew the percentage of phytoplankton that had food vacuoles, and then you had an estimate of the food vacuole cycle turnover time, you could get these rates probably about as good as you would by using some kind of incubation. And people did that like in the early days of just studying Bactivery. How do people try to use that method, but I haven't seen anybody apply it to mixotrophy.
So those were all the different things we discussed. It was a lot. We were bouncing all over the place. If anyone else wants to jump in and clarify anything that I said or add to it as well, like George helpfully did. All right. Becky?
Well, I think that's everything. I want to thank you guys for sticking around, but I also want to really thank you for participating in the first place. I think these were some actually kind of fun discussions. Makes us all think about mixotrophy again for a little while, every 2 months, if we're not actively writing proposals all the time.
I'm always thinking about mixotrophy. I don't know about you guys.
Yeah.
So yeah, so I'm really thankful for you guys for joining us.
Okay. Take care, everybody.
Yeah, just some very, very quick housekeeping items. Next meeting will be in November. The topic will be evolution, which will make Holly very happy. She's been pushing for that topic, obviously. We'll finally get to it. This working group looks like we will be continuing into 2023 because we've got money from all of the virtual or how virtual our meetings ended up being. So we should be good on that, and we have sent around those emails about that next meeting, and we are working to maybe nail down a specific date with that, but it will very likely be at Bigelow, not overlapping with OCB, but it might be close to OCB. And and Susanna Wilken asked if there will be another virtual writing session this week, and the answer to that is yes. The last virtual sessions will be this Thursday. For a variety of reasons, I've been very inconsistent with those, and I greatly apologize for that. But there will be one this week. And Becky.
Well, and that reminded me about the synthesis paper. We will get back to that. I met
So
with,
you are able to still work on that?
I believe so. I met with Natalie Cohen last week, and she gave me an update on the stuff that she gave to you guys at OCB. And we had a little discussion about where to go with that next. So I will be getting back into that synthesis paper. And the reason Nicole mentioned that, am I still allowed to work on that? I've just accepted a position as a program director in the Office of Polar Programs, Antarctic Organisms and Ecosystems starting today. So I become a conflict of interest with every one of you, or at least with this working group. So I can no longer be part of the leadership, but I'd like to thank everyone. It's been a really interesting and fun group to be a part of. I think I can still pop into your working group, these meetings, but I can no longer be part of the leadership. So
yeah, her orientation is at 1, so I guess it's like literally the last thing she's like allowed to do before it becomes an official conflict of interest.
Yeah,
stuck in under the radar.
So.
All right. Yes. Thank you everyone again for joining in on these meetings, and I'll see you in November. But you'll definitely be getting emails from me.
Good. Congratulations,
Bye, everybody.
Becky.
Thank you.
Thanks, George.
Thanks, everyone.